Good afternoon. I hope you had a gap in between and we can continue with the next session. So, yesterday we started with a work where we began our journey, and it is a slight continuation of the study what we did at the with another candidate. What I thought is that instead of explaining the techniques and all, let us study how the work we planned, how it was executed, and how we could finish off the work. And here you can see the title that. So yesterday we actually we worked on only on the natural products and in vitro studies and in vivo study. But here we want to combine the study with bioinformatics, and along with that, at the end we will be reaching the in vitro study. So sometimes maybe depending on your facility or depending on your strength, you have to plan the work. Sometimes it may not work in that way. So try to find out what works better for you. So in this case, so we did not work on the animal models, thanks to Dr. Nag, because this mice is from his lab. So we were studying the anti-tumor activity in the nude mice. And before going to our study, we will see why the in vitro studies, in vivo animal model, sometimes we hesitate to take that one for our research purpose. The major thing is that the time that consumes, okay, because whenever you are going for any in vivo study, it is just like handling the humans, okay, even they have the life and they also respond according to the way you respond to them, okay. So they need some time for growth, they need some time for, if they are having any disease, that time you have to take care of all the things whatever you take care of a particular patient, okay. So in that condition, it is lot of patience and lot of time. You will not get the result. Today I will put all the reagents into the mouth of the mouse, and tomorrow I will not get all the results, because it should take up the drug and later it should respond to the drug, because it has its own microenvironment in its the mouse body, and that should be metabolized and so many other aspects, and it takes a lot of time. And at the same time, not only that, the labor intensive. You have to run around, you have to feed them just like human beings, they need the food, and you have to feed them, you have to give them water, and you have to make them comfortable in all the ways, otherwise they may die, okay. So in such occasion, it is a kind of very labor intensive. You have to make sure that even the animal cages what you are using is clean, and every time you are changing that bedding material, everything, that takes you a lot of time, okay. And other aspect is that, along with that, the cost of the in vivo study, how much it costs to carry out this study. Because if you are using the Swiss albino mice or any other mice itself, it is a very costly business. So here even one mice if you purchase, it may be in Indian rupees, it may be around 100 to 200 rupees to get a one mice. But you cannot work everything with one mice, because you are supposed to keep the control and you are supposed to keep the drug, and in between you have to carry out all the toxicity studies and drug standardization, everything. And that takes lot of cost, because even the feed you have to purchase. They have a specific feed you can, or even you can prepare in the lab itself, but it is very lengthy process to make sure that it is a properly made and it kept in a sterile condition and all. It requires, the cost will be a bit high when compared to the in vitro studies. And one more disadvantage, it is a general disadvantage. If you do even in using animal models, you are not sure that it mimics everything in the human being, because the way mouse it has a maybe body, or its way of the interacting with the drug, or it may be its reaction to the drug may be different from how we are actually responding to the action of the drug. So naturally that makes a lot of difficulty for, that's why many drugs, even though qualify in the preclinical trials, they will fail in the clinical trial later. Okay, that is what the major difficulty is. You are investing so much amount and finally you will not end up with that. Or along with that, the other thing is that ethical issues. So you have to get the ethical permission, and along with that you have to maintain all the standards and regulations that is put forward by the ethical committee. So by crossing all these hurdles, if you carry out the experiment and later it may not completely predict what happens in the human beings. That's why we can check all those things in the animal study as maybe in vitro studies. So what are the advantages of in vitro studies compared to in vivo studies? One thing is that less expensive. You need not, maybe I am telling about not only the cell lines, there are other in vitro studies where you can cut the cost of the study when compared to the animal models. And somewhat it is easy when compared to animal models. And as I told, the animals, they need some time, because once you induce the tumor, tomorrow itself it will not grow into tumor complete size. You have to wait, you have the patience, and that will take you long time to get the results. But in vitro studies, you will get result much faster. I don't say it is completely better than the in vivo study, because in vivo study is much superior to the in vitro study. But the thing is that when you compare all other aspects, in vitro study is much easier to do. Screen large number of drugs, because when you take animal, you have to maintain certain animals, statistically it should be significant. All these parameters you have to check, and you have to check the drug on that one. So, and one more thing is that suppose if you want to check that drug further, you can reduce the number of the animals, so that the work, maybe the intensity or maybe the amount of work you are spending for in vivo studies, that can be reduced. And in another thing is that you can find out what way it acts on the cell. Okay, may not be completely, it may not mimic the microenvironment that is existing in body, but at least you can find out where exactly the drug is going and binding to the different regions in the cell. Whether it is acting on the enzyme, or whether it is acting on the maybe at the DNA level, or whatever it might be, you can find out that one. And along with that, even you can find out how far extent the drug has the effectiveness. So whether the drug has any toxicity or not, that can be found out. So along with that, nowadays even we have 3D cultures, tissue engineering, and that can simulate somewhat the in vivo conditions. Okay, so when you have that kind of maybe 3D cultures or tissue engineering, spheroid culture and all, you work on that one, and if you find a very good result, then you can move on to the in vivo study. That will be much easier. And then I will come on, maybe I will switch over to the topic what I was discussing yesterday. So we ended up with this result actually yesterday. So the Mr. Anand, he worked on the isolation of the compound, and once he isolated the endophyte from the medicinal plant, he purified the compound, and then we found out that this actually acts on the HIF1 translocation. It is inhibiting the HIF1 translocation to the nucleus. So we were curious, instead of isolating all these things, because it used to take a lot of time. We know how much time we spent to purify a single compound. So maybe we were actually carrying out the TLC and column, only with those two we were studying, and it used to take lengthy procedures and lot of time. And what we thought was that instead of taking the natural compound or anything, better let us go for certain other techniques where we can straight away go to the compound and then we can study. So the best that method what I found was that we can go for the bioinformatics method. Okay. So once again I coming to back to the institute where I was working at that time, that is around 2016 time, and that time Anand has completed his work, and we know that HIF1 can be the target for many of the molecules that are acting on the tumor. And that time the another person, another scholar he joined, his name is Jagadish. He actually, my way of conducting the research with the student is that first I will study what they are good at instead of what is my plan. So accordingly, I will try to design the plan so that they will be very comfortable to carry out the study in that way itself, instead of forcing something which is not working out with them. So I found that Mr. Jagdish, he was very good at handling the computer. Whenever I used to give him some work, he used to prepare a very nice slide, and the way he used to present, everyone used to get attracted. Then I thought that why, because that time also we had the power fluctuation and all other problem. Once the institute is established, maybe in as a new, it takes a lot of time to get established. And at the same time, whatever the UGC grant and BRNS grant we had had, that was actually getting over. So we did not had any new grant at that time. And the best way to come out of these kinds of situation was that we could work with something where there is no need of much investment. And at the same time, he got the UGC fellowship for doing his work. That was the one advantage. So I thought that better let him work on the aspect which he is comfortable, and at the same time I can plan accordingly so that his work will be much easier and we can reach the goal much faster. So what we did was that we thought that hypoxia would be the best factor for our study. So here briefly I will tell about what is actually it is. Hypoxic condition is a condition where there is less oxygen. So this condition actually makes us to feel uneasy. Suppose if I block all the windows here, if the oxygen concentration goes down, naturally you feel very uncomfortable, uneasiness. And we know that even if there is any building catches fire, people not die because of the fire, they die because of the carbon dioxide and carbon monoxide. So once the oxygen concentration reduces, person is prone to die. And at the same time, this hypoxia is related with many aspects. Maybe some of you might have observed also, even in anemia there will be hypoxic condition. The person will find difficulty to breathe because the less RBC cells, he cannot or she cannot get proper oxygen to the body, and all the symptoms of hypoxia can be observed. Or sometimes if you go to the high altitude also, there is a lack of oxygen, that time also you find this hypoxic conditions. Or deep down the sea, if you go, there also the oxygen concentration will be very less, you suffer from, or if you have not proper the aids, you will be having hypoxic conditions. So all these conditions, maybe that is maybe generally what it happens, but what happens during the cancer, because we are relating the hypoxic condition to the cancer. So we studied about this one, and here if you see the picture, the picture what it shows is the cells that are dividing continuously. Okay. So as the cancer cells, they are getting divided and they start getting accumulated, and as they getting accumulated, they are far away from the blood vessel which supplies the nutrients and oxygen. And as a result of this one, the cells which are far away from this blood vessel, I am not going to tell you exactly how much distance, what way, what happens and all. So if they are moving far away from the blood vessel, what happens is that these cells becomes necrotic cells. And these necrotic cells, they will not be having that kind of oxygen. And at the same time, this condition actually induces the factor called as hypoxic factor. That is called as hypoxia factor 1. Or it is alpha and different factors, they are there. And at the same time, as the blood vessels, they are far away, this cells, they actually suffer from oxygen concentrate, maybe oxygen supply, and they become hypoxic cells. And the hypoxic cells, they try to adapt, because whenever we are under stress, usually we try to adapt in one or the other way. The same way, because here the major problem with the cancer is that they are part of our cells only. They are actually kind of misguided or maybe misoriented cells. So that's why it is very difficult to treat the cancer when it comes to, except few cancers. I am not telling all the cancer cannot be treated with that one. There are certain cancers difficult because they are actually kind of maybe they are origin from our own body. And whatever we treat the cells or treat the cancer cells, same thing is taken up by even the normal cells or non-cancerous cells. And here in this case, what happens is that the oxygen gradient, it goes on decreasing as the cells are away from the blood vessels. And at the same time, if alpha factor, its concentration increases because hypoxia induces the factor called hypoxia 1 alpha factor. And at the same time, these cells, they switch over or they undergo metabolic reprogramming, what we call it as. So metabolic reprogramming in the sense, they go for more production of this lactate dehydrogenase, and lactic acid gets accumulated. Those who have studied biochemistry, you will be very much known about, maybe very much aware of this one, what happens. And as a result, the acidity or hydrogen the concentration increases or pH decreases. And at the same time, even the carbon dioxide gets accumulated and it contributes to the pH gradient or the less pH activity. And the cells, they switch over to the glycolysis because there is an accumulation of the lactate. They cannot move to the this complete, they switch over to the anaerobic and become glycolysis, and this makes the cell radio and chemotherapy resistant cells. And this makes the very difficult to treat with chemicals or even with radiation therapy, because it will not able to bring about the effect that much effectively when compared to the condition which is prevailing in the normal condition. And here this it has been shown since many years that this hypoxia is the one which actually hinders the therapy since many years, and we were studying what best we can do with this hypoxia factor, because we planned better let us work on the hypoxia factor. And then we wanted to study its structure so that we can move further, because earlier study it was very simple. We know that the plant was anticancer, and we knew that we can isolate a compound that is which is a lead molecule and we can go further. But here all of a sudden we ended up with a molecule which plays a role in the cancer. So we were stuck there, and then what we did was that just we went on a simple study. Only one thing what we did was that we were answering the question why we have to do this one. So we wanted to study, because if you have to study the hypoxia factor, you should have a knowledge about the hypoxia factor structure. This is a factor, it is actually a transcription factor. This transcription factor, once it is a transcription factor, most of the biological science students, you are familiar that it induces the expression of some other proteins. In simple terms, I can tell that one. Okay. Just like the human body, even the transcription, because I compare that whatever happens with that molecule also, they are kind of a humans. Here this, the transcription factor has different regions, like BHLH, we call it as domains. Okay. And even the past domains, don't worry too much about the domains and all those things, just see the structure of this one, because we were actually going to study all these aspects with this structure of the molecule. And then we saw that it has a oxygen dependent domain. Oxygen dependent domain is the one which determines or which senses the presence of the oxygen. Suppose if the oxygen is present, strange thing is that this hypoxia factor is degraded within 5 minutes. Suppose if the oxygen is absent, it becomes stable by interacting with other molecules. And also it had N-terminal domain, C-terminal domain. So we call this past domain as dimerization domain. So this actually favors the interaction of this hypoxia factor 1 alpha with other protein, which plays a crucial role in bringing about the cancer. And another one is that regulatory domain. Domain in the sense, those who are not from biological science background, it is a region where it is involved in certain functions. Okay, if you take any protein, let me consider if my body is a complete structure, my hand is a kind of one domain. Okay, my leg is a kind of one domain which performs independent function. That, if you remember, it is more than enough. So another one is that carboxyl terminal domain. All this domain, they are present. So what we should do next, because that's why, just to show that what are domains, I have taken different organs and they are doing independent work. So we wanted to know then whether there is a single hypoxia factor or different other factors are also involved. That time actually many this hypoxia factors, they were getting reported. So they were having different functions and they were getting reported, because we wanted to use according to our limitations, according to our strength. We did not worry too much about what is happening, because our limitations made us to think in that way. And there are different factors we noticed, that is HIF1 alpha. Along with that, there are other factors were also expressed. They are called as hypoxia factor, like 1 beta, 2 alpha, 2 beta, 3 alpha, 3 beta and all. So they had different functions and they have been reported. But we were not that much interested about the other factors, because of we were targeting HIF1 alpha. And then what we did was that because what we observed in our previous study, that this HIF1 alpha, actually it is involved in so many functions, and it is straight away it is linked to the cancer. We wanted to find out with how many protein actually it is interacting with. So we checked this interaction of the HIF1 alpha with all other protein. We could see that it is interacting with so many proteins that are involved in the cancer. And it is just like us, we will not interact with only one person. With simultaneously you might maybe interacting with different people, and for different topics you might be interacting with someone. Even the if this hypoxia factor, it is interacting. It takes the signal from one molecule and it actually sends the signal to the other molecule. The same way what we do, they have their own network and signaling. And we wanted to find out, because this actually we wanted to study so that we can plan our work properly. We studied what happens to the hypoxia factor during the normal oxygen concentration and during hypoxic conditions. So during the normal condition, strangely, maybe within 5 minutes, this HIF1 alpha factor, it is bound by one enzyme called as proline hydroxylase enzyme, and it brings about the hydroxylation. Hydroxylation is, in the sense, it is just the addition of hydroxyl group. And that hydroxyl group is recognized by some other group of protein that, let us not worry the complete about what type of protein they are interacting. And ultimately what happens is that this protein is chopped off into smaller pieces. That means in the normal oxygen concentration, it is getting degraded just within 5 minutes. But when the oxygen concentration is low, it is bound by some other protein. It is just like you are having a kinds of friends where when you have enough money, you have some friends, and once the pocket is empty, you have another friend. So like that, when the oxygen concentration is high, the friends are different, even the protein factors. And here, once the oxygen concentration is low, naturally it is bound by different factors, like even the radiation causes the activation of this one. And that makes the protein to bind to certain other proteins, like the other stabilizing protein. I don't go in detail about this different factor because it may not be familiar with. There are some chemistry people are also sitting, they may find it very difficult. So here they are also binding to some other protein, and what happens is that instead of getting degraded, they are actually become stable. And once they become stable, they are bound by some more proteins. And these proteins together, they enter into the nucleus where they bind to the specific region on the DNA, where it brings about various other aspects which I have listed out here that is linked with the cancers. You can see that metastasis or angiogenesis or Warburg effect or even pH homeostasis or cell survival. So many different aspects that are brought about by one signaling molecule. That is the hypoxia factor. And next we wanted to study the sequence scientific, because there were already reported there are different the hypoxia factors are there. Best way is that if you know a person like Joshi, if you find there are 100 people here Joshi, best thing is that you find out the similarity between them or differences between them based on the height, based on the campus, or based on the different aspects. We did the same thing. We wanted to know, differentiate, or we wanted to find out the similarity between those, and we found that there are some similarities and there are some dissimilarities. I am not going in any detail about that one. And next question was that, because this is a very strange thing what we observed. We were checking, we were very curious because we were asking some few questions why. Usually people, they are getting cancer. Is there anything that doesn't get the cancer was why our major question. So anything that usually resists the cancer was the question. We started searching the literature. And as we were going through the literature, we got an article which I have quoted here. It shows that there is a mouse. It doesn't look like a mouse, but it is a mouse only. It is called naked rats because they don't have hairs on them, and they actually live in the burrows, and they are actually present in a very hypoxic condition, and they live longer than the mouse which are growing in the laboratory as well as even in outside, regular mouse. And then we were very much interested because the cancer and the hypoxia may not be related in that case, because always we say the hypoxia is related with the cancer. And at the same time, there are certain organisms, they don't have cancer, but they have the hypoxia. Okay, it is a contradictory kind of the results what we were getting. We were stuck at that time. And then we studied what is the mechanism by which actually it evades that the cancer. So there are certain reasons where it produces the hyaluron, that is a kind of compound which is present in your knees where you can move your leg properly because of that lubricating kind of the polysaccharide substance. So same thing, a derivative, a large molecule that is present, that actually prevents this, this naked rats not to get the cancer. Whenever the cells from this, the naked mole rat was grown in the laboratory, they used to produce lot of this, the hyaluron, that is high molecular weight hyaluron, and the cells used to be stick with each other. That's what they use a particular terminology called as goo. It used to produce a goo, and cells used to stick together to each other. And when it was happening like that, and they wanted to, for a steady sake, what they did was that they removed all this, the hyaluronic, hyaluron, and then they found that the cells were prone to the cancer. That means that even if they are in the hypoxic condition, they are not susceptible to cancer because of this particular heteropolysaccharide that is present. And not only that, we came up with another article saying that there is one more rat that is actually highly resistant to the cancer, and it lives longer compared to the other normal mouse. So that is called as blind mole rat. Okay, and here the mechanism is totally different when it comes to the blind mole rat. It is also highly resistant to the cancer, but because of the presence of DNA methyltransferase 1, it can prevent the cancer spreading. That's what the article says. And these two are the organisms which we were got the information that they are highly resistant to cancer and they live longer. One is the naked mole rat, and another one is actually the blind mole rat. And then we had a question, because if you look at the conditions, the oxygen concentration is less even in depth of these oceans and at the high altitude also. So we will carry out a study where we will study the relation or maybe the correlation of this hypoxia factor in different organisms. They are they are living in different atmosphere, whether it throws any light on our study. So we wanted to select the organism. Our idea was very simple. We selected few organisms which are related to the human being, because we were looking for the bioinformatics study. So we selected the organisms like human, chimpanzees, maybe this orangutan and monkey and all. And along with that, even we selected dog. And at the same time, the other animals we selected, whether it is cow or yak, which are in the maybe Tibetan antelope, which is actually living in the high altitude, to get an information. And at the same time, we selected even the lab mouse also, okay, regular normal mouse, and even the rat, and we included these two, because earlier study we did not included these two, because we did not know that there are certain organisms, they can survive even at very low concentration of the oxygen. So we selected that one, and then we selected the two organisms. They are surviving in the water. They are actually having the hypoxic condition there. So our intention was very much clear. We have taken almost all the organisms. They are representing the different atmosphere where the hypoxia factor or hypoxia factor is getting expressed. So once we studied that one, we searched the sequence of the hypoxia factor of these 16 organisms. And the best way to compare them was that, because we did not had any this lab to check the in vitro or in vivo type of studies, what we did was that better let us study with the computer itself, whether they have any relations or not. So we found that best way is that you can carry out the multiple sequence alignment. Most of the research scholars, you know what is multiple sequence alignment. You just take the sequence of the DNA, you match with the other DNA, you find out how much extent they are similar or dissimilar. This is the basic principle. And we found that there are certain differences, there are certain similarities. I am not going in detail, the technical aspect. We found that there are certain variations in certain region, and there are certain maybe similarities in some organisms. And these results, even we took, there are certain domains also, and we saw any this if there is any similarity and dissimilarity, and we found that we could arrange the organisms according to the hypoxia factor, the relatedness with the hypoxia factor. If you look at the human beings are related with the chimpanzees more, and they are related even with the orangutan, with maybe with related to hypoxia factor I am telling, okay, nothing else about the evolutionary history. Just I am telling with respect to hypoxia factor. And even we could see that all these yak, cow and Tibetan antelope, goat and all, they belong to the same category. And this blind mole rat, and interestingly you can see that the blind mole rat and mouse, they are at the one category, and this deviated, this naked mole rat, it deviated with its relation or similarity when compared to other. And as expected, this naked carp and zebrafish, they were in the separate clade. What does it indicate? So this indicates that it is a kind of evolutionary relationship between the hypoxia factor, just like the humans evolved, along the way even the hypoxia factor evolved. And then we wanted to study, because I was telling about a domain or a hand which is dependent, or its stability or instability is dependent on the oxygen concentration. We wanted to find out how far extent this oxygen dependent domain is related with each other organism. You can once again, you can see that they have certain relations, and there are certain organisms, they moved out of the clade, and there is a relationship between even the oxygen dependent domain also. And then was that we wanted to find out, because the blind mole rat, which is a highly the cancer resistant, or even in hypoxic condition which cannot get the cancer, and the humans, we are naturally prone to the cancer. And this HIF1 alpha domain of these two organisms, do they have any relations or not? We found that there are certain key residues which I have marked here, they were one and the same. And there are certain residues where there was a difference between the humans and maybe this blind mole rats, indicating that maybe these residues may be playing a role in the way organism it responds to the oxygen. So because the candidate was enthusiastic, and this study seems to be very simple, but it took nearly around 6 months to do all these things, because he was supposed to learn all the bioinformatics tools. And then we came up with a paper saying that what is actually the sequence analysis and phylogenetic studies of hypoxia inducible factor 1 alpha. And then what we did was that now we know that we are very much sure. So there is a relationship, and there is a dissimilarity between the organism with respect to the hypoxia factor. Then what we approach this hypoxia factor? Can this hypoxia factor be the target of anti-cancer drugs? Because we did not know which drug will actually bind to the hypoxia factor. So for that, we started using the bioinformatics. Instead of taking one molecule, and at this end, if you end up with none, it is waste of time for even the research scholar, and it is waste of energy. So what we did was that instead of doing like that, let us create a maybe in virtual condition where the hypoxia factor will be selected, and a drug will be selected in such a way that it will go and bind to that one, and it prevents its interaction with other molecules. Because earlier itself I was telling, the hypoxia factor is binding to the different proteins, and those proteins are the one, they will determine whether this hypoxia factor to be degraded or it should be disabled. So that's why we wanted to study, is there any molecule which can prevent this protein-protein interaction, so that we can target that one and prevent the stabilization of hypoxia factor, and in turn we can treat the cancer. That's what our hypothesis was. And for that, what we were supposed to do, I am simply telling about what actually we did. I am not telling each and every technique what we are doing. And we took the receptors, because receptors also we are getting maybe we have to build the models, because unless if you don't have the model, you cannot bring the drug and you cannot target it to the models. So we were supposed to build the model for that hypoxia factor. So we took the PDB, protein data bank, and we modeled using various softwares. And not only that one, there are different ways we have to authenticate that one. So either it is pre-processing or grid generation and all, let us not worry too much about that one. So ultimately what we did was that in our study, we prepared a model which can be docked. Docked in the sense, I can bring a drug and I can put it onto it, and I will study whether it interacts with the other proteins or not. So we did that one, and even the ligands, if we have to select, they should be selected in such a way that there are certain rules we are supposed to follow. They should have certain amount of size, that is, we call it as Lipinski rule of 5, and if they actually qualify that one, then we will go ahead with that energy minimization, and all other studies we will do. It is just like the way, if you start writing, you need the pen, you need the paper, you need something else, you need the chair, and if everything is proper, then your study goes fine. So if the chair is not proper, you will make sure that that chair should be proper. In the same way, we made sure that different techniques we have used, and we made sure that whatever you are doing is foolproof. And then we analyzed, and later on we actually visualized the molecule, how it acts. And we selected different domains, that's what I was telling, various domains, and we have used different the software packages to consider this PASS domain or BHLH domain or CTAD domain. Don't worry too much about this one. And then even we studied whether it is the dope or dope score model or even Ramachandran model to establish that it has a proper way of arrangements. And finally, we wanted to target HIF1 alpha factor. And in HIF1 alpha factor, as I was telling, it has different domains. So whether I should target that domain or this domain, we had a doubt. So we went to the literature. When we were studying, we found that the PASS-B domain of HIF1 alpha is majorly involved in the interaction with the HIF1 beta. And to become along with that, the PASS-B domain, it also interacts with another protein that is called HSP90. Most of the biology students, you are familiar with this HSP protein or heat shock proteins. They are actually considered to be the molecular chaperones. They maintain the structure of the protein. Okay, these two proteins, they were actually interacting with the HIF1 alpha protein. So here, the HIF1 alpha beta interaction is necessary for the entry of the hypoxia protein into the nucleus and bring about the activation of many genes which are responsible for cancer. And the HSP90, it actually stabilizes in such a way that the HIF1 beta can bind. So these two factors, actually the key factors, they are making this HIF1 alpha a strong molecule so that it induces the cancer. So then what we did was that we analyzed what are the binding site, because if you shake hand, you know what are the regions actually interacting with one another. Just if you put some block between the hands, then naturally there won't be any interaction. That's what our the plan was. So HIF1 alpha, HIF1 beta interactions, they are mediated through some key amino acids. So if you can block the interaction between those key amino acids, naturally there won't be HIF1 alpha, HIF1 beta interactions, and naturally that the HIF1 alpha is taken away by the degrading enzymes, and it is completely degraded. So then we wanted to dock with compounds. That's what I was telling. Instead of taking one molecule, why can't we select too many molecules or library of compounds and we screen that one? Because we did not had the facility, we can't sit quite. So we had the computer and we could connect it to net and we could do some studies. We thought that better we will go ahead with the library screening. So we selected nearly 67,823 compounds, and then we applied the Lipinski rule of 5. I am not going in detail about all these rules and regulations. They actually, one thing you remember, this rule implies that if the 5 rules are followed by a compound, that compound is considered to be the drug. It should have a molecular weight between 500 Dalton, something like that, or it should have a capacity to cross certain region, all those things. And this led us to the key down to around 2,753 molecules. And later on we actually further screen the compound, make sure that whatever we are doing the correct study, and we created a compound library, and this compound, from the compound library, a pool of these compounds we have selected, and we wanted to dock and we wanted to find out, and then we carried out the docking study with various softwares what I have mentioned here. And this thing is that actually Schrodinger, we had an advantage. At that time only this Schrodinger software was purchased in the Department of Industrial Chemistry, and we actually took the help of the industrial chemistry to carry out this Schrodinger, maybe to carry out this docking study and other studies using Schrodinger, and we could get a better result. And when we analyzed this result of the compound library what we prepared, we found that there are certain compounds. Usually whenever in the bioinformatics we have a practice, we will study what is the binding energy. The best way to find out whether one molecule will bind to the protein in a proper way, whether it interacts with the proper way, best thing is that you go and study what is the binding energy. So if the binding energy is maybe it is shown in the minus, if it is the minus value, if it is high, then it indicates that it has a very high affinity to its target. So we studied nearly, here I have mentioned about 13 compounds, and these 2 compounds we found they are attractive when compared to other compounds, because we cannot carry out all the studies using all the parameters, because there are different hiccups between them, because each work it takes a lot of time to do that one. So then we docked it and we studied whether the molecule really it is going and binding to the drug. And we found that of course they are interacting with the drug, and they are interacting at different regions of that particular protein. So I have mentioned here different amino acids. They are the key amino acids. They are actually interacting with this particular compound. We called it as D1. This is the compound. And later on, the same thing that we studied with another compound, that is actually the 2 compounds we selected, and this one was also actually binding to PAS-B domain. So PAS-B domain, as I told earlier, is the domain which is necessary to interact with the HSP protein as well as HIF1 beta. So if I can show that it is binding to the pass B domain, it is indicating that it is preventing the binding of the other protein. It is just like if you are holding one hand with maybe your friend, at the same time you cannot hold the other person's hand. So same thing we assumed, and we thought that these two are the better one for further studies. And later on, we did not stop there, because one is that we wanted to find out what way this HIF1 alpha and HIF1 beta interaction can be blocked. On the other hand, we wanted to find out how HIF1 alpha interacts with the HSP90, because HSP90 is the one that actually it mediates the stabilization of the HIF1 alpha factor. So we wanted to find out what exactly that interaction is altered by our molecule, and we did the similar studies to find out what way it actually prevents that the HIF1 alpha HSP interaction. We found that there are different molecules, they are also preventing this HSP as well as HIF1 alpha interactions, that is the protein-protein interactions. And these are the studies that are showing how exactly these molecules, they are interacting with the protein. So there are different compounds, I am not going in detail about that one. And when we were studying this one, the advantage for me was that if I tell one word to him, he used to find out 10 different possibilities of that work. And he was actually studying, we were actually focusing only on HIF1 alpha, HIF1 beta and HSP90. He wanted to find out what is the effect of this particular protein on different other protein. That time there was a new protein that was developed or maybe reported, that was called as Tigar protein, that is TP53 receptor protein. And that protein was actually involved in the oxygen gradient and all. And that is somehow linked with the cancer. He got a paper and he was very curious to know what way this hypoxia factor that actually regulates that protein also. So he started studying about the, once again we constructed the model and we wanted to find out what molecule that binds to the molecule. And later we wanted to find out, because this Tiger protein, it actually interacts with phosphofructokinase B2, and we wanted to find out how they interact. I will not go in detail about all these aspects. And later we did the simulation study. In bioinformatics, one thing is that whatever you predict may not be right. You have to mimic the condition that exists in a natural way. That we call it as simulation studies. So once we did the simulation study, we found that this protein also interacting with the HIF1 alpha. With that one, we could propose our hypothesis saying that how this Tiger protein with the maybe hypoxia factor, they can be the key regulator in apoptosis regulation, and it got accepted. So that means our hypothesis, we assumed that it is right. And then once we knew that there are certain compounds, they prevent the protein-protein interaction, and we got the information from our study that they can be the best drug. And that we wanted to cross-verify. The easiest way was that the cytotoxicity assay, because that's what we did. So here we usually, those who are not familiar, because there are some MSc students, yesterday also I mentioned what is cytotoxic study. We will take the cells and we will detach these cells which are grown in the culture, and we will take in a plate which contains, we call it as microplate, and we add the drug and we incubate, and later on we find out the absorbance and we will find out what is the action of the drug. This is the simple theory behind cytotoxic assay. And when we did that one, we were thinking that if it is supported by our whatever we did in bioinformatics by the cytotoxic study, then we are right. So we checked with different cells, and then we had a question, which cancer cells to be selected for these studies, because I cannot randomly select the cancer cells and I cannot say this is acting on HIF1 alpha. So because I wanted to show that the hypoxia factor is increased and it prevents that one, and even at the same time it prevents the cancer. So we searched the literature, we found that there are certain cells where hypoxia factor is produced a lot. Maybe one of that one is that breast cancer, as well as even in the prostate cancer, we found that the hypoxia factor is expressed in very high concentration when the tumor is present in a patient. So we thought that let us select the same cancer cells and we will find out the cytotoxicity. Of course, we got the cytotoxic activity of the drug, and when we found, when we compared this one with the non-cancerous cell, it was not showing that much activity. That means it is not that much toxic to the non-cancerous cell when compared to the cancerous cell. The same thing happened with the other drug what we did. That drug also showed a similar trend where it was active against the cancerous cell, but it was not active against, maybe lesser active against the non-cancerous cell. And I told you, because we wanted to show that experimentally that it is actually acting. Just we wanted to show that the molecular mechanism also, that it is preventing the HIF1 alpha, HIF1 beta interaction. So we wanted to find out how exactly it is actually preventing protein, maybe alpha beta interaction. We clearly showed that whenever, and here one more thing, we had a problem with the generating hypoxia for the cancer cells. But we searched the literature whether we can do any other method, because hypoxia chamber is costly. We could not afford that much. So we did not have the fund. We wanted to find out, is there any alternative way we can artificially create an atmosphere where it mimics the hypoxia, which creates the hypoxic condition. And we found that this cobalt chloride could induce the same effect, and we used that one, and we studied what happens to the HIF1 alpha expression if we treat the cells. And we found that whenever we treated the cells with the D1 or D2 drug, we found that actually the expression of the HIF1 alpha, it is coming down, indicating that whatever we hypothesized, that is actually working out. And then even we did the immunofluorescence study. It looks blank, but in my computer it is showing, it is not a good picture. And here also the immunofluorescence study, we showed that whenever there is the drug D1 and D2, it is actually preventing that the interaction of the HIF1 alpha and HIF1 beta. And based on our study, we designed a model. What is actually the mechanism by which the drug is acting? And here we found that the drug, whatever we had selected, this prevents the HIF1 alpha, HIF1 beta interaction. These are the two drugs. And there is another drug which acts on, we studied even the tiger, and it acts on this particular region. And the other 4 drugs, they are acting the interaction between the HIF1 alpha as well as HSP90. So all these suggested that suppose if we block that HIF1, maybe the protein-protein interactions, that protein-protein interaction definitely has a good role in bringing or reducing the cancer. That's what our hypothesis. And then we studied further, because if we want to continue that study, we studied with various other aspects. Just I will, for your sake, what are the limitations of